We've established a little bit of a pattern here where I start with kind of more the background side of things, bring us up to where Ed's group joined, and then getting into the study that we'll talk about today. That's what we're planning to focus on anyhow, is the actual study that you just mentioned. So I'm going to try sharing my screen here. There you are. And then go to the appropriate slide where I've greatly simplified the title of our paper. Maybe not greatly, but I have simplified the title of our paper. And I want to start by just really briefly at WSU. So there's the Washington Stormwater Center, and it's jointly led by WSU and UW, which is awesome that it's almost a coincidence actually that that we're working on this study together, but we both fall under this umbrella of the Washington Stormwater Center. So for my side of it, what my research program focuses on is understanding how stormwater impacts aquatic organisms, how toxic it can be to different aquatic organisms, and in what ways we need to be concerned about stormwater. And then also looking at possible solutions for that toxicity. So that's sort of the context background for where I'm coming from with this research. And of course, many of you are aware that this work has been supported over the years by many, many different partners and various funders, most notably NOAA Fisheries, U.S. Fish and Wildlife Service, and then also the Suquamish tribe played a large part in getting us where we are today, and also the Puyallup tribe more recently. And a lot of our funding has come from EPA Region 10 directly or indirectly. And we've also worked with many of the other groups on here, including the Stormwater Action Monitoring Group and the state directly. So I think everybody's aware of this, but it's always good to start with a really tragic video of a gorgeous wild fish succumbing to stormwater runoff. So this is a Coho salmon. And you can see he's still very ocean bright, right? Just barely starting to turn those spawning colors. This is not what should be happening to this fish. So it's about 20 years ago that people first started documenting this phenomenon, first started studying this phenomenon. Anecdotally, we have evidence, from back in the 80s that this was happening already then, and possibly earlier than that, because the simple truth is that we expect to find dead salmon in creeks in the fall, right? It wasn't until people started noticing these abnormal behaviors with Coho returning to restored creeks that we started opening them up to see whether or not they had in fact spawned. And it turns out that Coho salmon returning to urban impacted basins throughout the Puget Sound, which is where we've been studying this. Not to say it's limited to here, but we do find that high rates of these fish don't get a chance to spawn, and it's both, it's recurrent and pretty widespread as well. So the first paper that we were able to publish on this was a bit of a forensics analysis, trying to identify what could be causing this. And we know that salmon die prematurely when they come back to fresh water to spawn for a variety of reasons. There can be, for example, sockeye salmon in the Fraser River system, and also Chinook in the Klamath River system, where we know that water temperature exacerbates disease conditions and that sort of thing. So, but we were able to rule out in these Puget Sound urban impacted creeks temperature as a correlate, disease as being involved, nothing wrong with the dissolved oxygen levels in the water, no one contaminant standing out, in terms of what we typically measure as being likely to be responsible for this. So that led us towards the stormwater runoff, and then, like was said, ultimately to road runoff. So the other thing that we learned early on is that at these rates of pre-spawning mortality, it's not difficult to predict extirpation, local extinctions of Coho salmon in these basins. And we find that even on the metapopulation level, even if it's just individual subbasins that are affected, it can draw down the resiliency and lead to potential extinction, even at that metapopulation level. So we've certainly been looking to Coho salmon as being a sentinel for stormwater impacts throughout this region. Of course, they're very widely distributed. This is showing all the little creeks where we expect to find them. And most of these are kind of lowland streams, right? Which is where they run into a lot of problems with us, because we like to build our developments in lowland areas as well. They also, unlike some of our Pacific salmon, they spend a good amount of time in freshwater, at least a year rearing in freshwater after they hatch, instead of just heading right for the salt. And we know that there are populations listed under the Endangered Species Act, either as a species of concern or in fact threatened. So like I was saying, we related this to stormwater runoff. It's not hard to imagine that stormwater could be causing a problem to some organisms because, of course, this is an extreme example of something that is not a forest. And we know that when it rains on this landscape, the water doesn't have anywhere to go. It has to run off. And so compared to systems that are less developed, you're getting a lot more of the precipitation that falls on these impervious surfaces having to run off into surface waters. And as it does that, it picks up all sorts of things, right? I love this video by Laura James, who's a diver who discovered stormwater runoff as an actual, she thought it was literally a column in the water that wasn't there before, went over to investigate it and then ended up, getting really involved in stormwater pollution. So here we're seeing the particulates that get carried with that runoff, but we're not seeing the chemicals that get carried with that runoff, many of which of course are invisible to the naked eye. So in terms of trying to understand now, okay, something about stormwater, what is it in stormwater? Of course, we tend to measure a simple suite of contaminants. We measure things like metals, sometimes we measure hydrocarbons, bacteria, nutrients, a few various other things, including pesticides. But we've learned over the last 5 years even, that there are thousands of chemicals carried in stormwater runoff. Bowen Du, who was a postdoc working with Ed, made that discovery using the high-res mass spec that Ed will certainly be talking about. We also know that relatively few of these thousands of chemicals are very well characterized in terms of toxicology. And we did try to make initially synthetic stormwater mixtures with metals and hydrocarbons, and could not recreate this pre-spawning mortality whatsoever, even at very high concentrations of metals. So one thing that we did learn through land use analysis, where we were showing, this is a predictive map of where we expect to find different rates of pre-spawning mortality for coho throughout the Puget Sound region. This land use analysis conducted by U.S. Fish and Wildlife Service and NOAA highlighted that not just development generally or impervious area generally, but more specifically, higher rates of pre-spawn mortality are associated with roadways and busier roadways at that. All right, so this is where we got down to that. And I put at the bottom here, feel free to email me if you'd like this information, but a link to a story map that we built to describe the pre-spawning mortality phenomenon and help maybe citizen scientists participate in documenting where it is in the landscape. All right, so it's been about 10 years now that we actually started collecting roadway runoff in order to do controlled studies, and that ultimately led to where we are today. So in the early days, it was just me collecting runoff off of this highway initially. Here's a sample of what that looked like. Just a convenient place to collect roadway runoff and start to learn something. We started working with the Suquamish tribe, exposing adult salmon recently returned from the saltwater. And so they're in these PVC tubes where they can remain calm, because this is actually a big wild animal. And this is a great way to work with them. So here we've exposed them for just a few hours to either well water or that collected roadway runoff. And if you get just a little roadway runoff, it just looks like water, but if you get a bunch of it together, it can end up looking pretty dark. All right, and then after a few hours, we can take those fish out and see what's going on. So control fish here is looking a little alert and awake after having had a nap in that tube. The fish that was exposed to stormwater for that amount of time is showing these very advanced symptoms we associate with the pre-spawning mortality. So we found that using actual collected road runoff, we could recreate the behavioral symptoms we saw in the field and also that rapid mortality. And we've shown since then that not just adult spawners, but that we also can cause this acute mortality with this water source for recently hatched alevin of coho salmon and also in juvenile salmon. We've also learned that much earlier than mortality, which is fairly rapid. It can occur within just hours, but even within like the first half hour of an exposure, we see behavioral changes. And so what leads to that complete loss of equilibrium starts out as a rising to the surface of the water and swimming. And so we've been able to document that with juvenile coho, showing that it's the same process as in the adults, which allows us to do a lot more work along the rest of the year instead of waiting just for fall when the spawners come back. And we've also seen that very early in that behavioral sequence, we know that the fish cannot recover. So it may take them hours to die, but in fact, the molecular events that have triggered that mortality happen very early. We also know that there are blood changes that are associated with the behavioral symptoms that then lead to mortality. Osmoregulatory imbalance is part of it. And then hemoconcentration, the blood getting very thick, appears to be a very important part of that. All right, and then let's see. So at the Suquamish tribe, chum salmon come back at the same time as coho. In many of our area streams, those two species overlap, and so we had the opportunity to early on to test another species of Pacific salmon alongside coho, which clearly showed this sensitive response. And this figure is summarizing that in the control coho, after a few hours in clean water, all the fish were fine. Maybe one fish was a little lethargic. In a few hours in the runoff, the coho are showing a variety of these behavioral symptoms that lead to mortality, including loss of equilibrium. In the chum, even the chum exposed to runoff look completely fine. All right, so the behaviors weren't triggered by the runoff exposure. We also saw there were no changes in the blood chemistry, and certainly there was no mortality even with 24-hour exposures in which all of the coho were dead. So in a way now we have a negative control with chum salmon. We've learned that it takes a lot of dilution of roadway runoff in order for coho not to be affected and not to die even. This is just about mortality, not about other sublethal effects that might be happening. So in fact, it wasn't down until we only had about 2% runoff left in our dilution that the coho all survived exposure. Even at just 5% runoff, so 95% clean water, we're seeing mortality. All right, so getting closer to what is killing the coho is this study that was led by Kathy Peter, another of Ed's postdocs, where we looked at a variety of chemical profiles from a variety of sources of vehicle pollution to roadway runoff, right? Things that are coming off of or out of our vehicles, and then comparing those chemical profiles to waters that we know kill coho. So the roadway runoff, and also various urban sites where we know coho salmon have died from exposure to those waters. And this analysis, this hierarchical cluster analysis, groups these different waters and sources of pollution by chemical similarity based on these chemicals here with all the colorful bars. Blue means the chemical's basically not there. More and more red means higher relative amounts of whatever chemical that is. And when you compare those across these waters and these vehicle sources, you know, all of the waters from the highway are more similar to each other than anything else. Same thing for the urban creek waters, but it's the tire particle water that's most similar to these waters that killed coho. So from here, we started focusing on tires, whether they're an important source of this toxicity that we see. So what we did is compared the response of coho to stormwater runoff with the response to a leachate made using tire particles. And if tires are an important source of this toxicity, we would expect it to cause acute mortality in coho. We would expect coho to be sensitive, uniquely sensitive compared to like chum salmon, right, which were not sensitive to stormwater, and also to have that same pathophysiology, the same changes in behavior and also in blood chemistry. So I'm going to summarize that study here quickly, and then I'll pass things over to Ed. So for this work, and this is some of the same things that we did for the science paper in terms of generating tire powder. We used a lot of graduate students and also some technicians, a carbide grinding disc on an angle grinder with a dust shroud attached to a shop vac, collected these particles. And ultimately we had 9 different tires. We weren't interested in this point as at this tire versus this tire. We were just interested in tires generally that might be on the road. So we ended up using 7 used tires and 2 brand new tires, one kind of high end, one kind of low end, trying to just get a wide variety of tire. And then we mixed these powders all in equal weights to get our fabulous 9 tire mix. All right, which we then created a leachate with by putting in these felt filter socks and having water run continuously through the particles in that sock for almost 24 hours. So this shows that setup, and we did something much more robust and fancier for the study with focused on the chemistry. All right, so the setup that we have with the Suquamish tribe gives us 4 different treatments we can do. So we have a control and then 3 different concentrations. And we did 24-hour exposures. And that first question, would a tire leachate cause this acute mortality in coho? And the answer is yes. So both juveniles and adults appear to have very similar responses. We've got this nice dose-response relationship where concentrations above like 0.3 grams per liter killed all of the coho. So in fact, we use that concentration for the future studies that I'm going to be showing you. And Ed's team has shown that this lower range where we're seeing this mortality here is associated with, or is reflective environmentally realistic concentrations in roadway runoff and or surface waters. Okay, so we use that 1 concentration now for the rest of these experiments, where we have tanks with 2 coho and 2 chum, because we were using the chum as that negative control, well water versus that tire leachate. And again, a concentration that we expected would kill all of the coho in 24 hours. And if tires are an important source of this toxicity, we would expect chum not to be affected. We repeated this experiment 4 times and got these really complicated results here where all the coho died like we expected. None of the control fish essentially died. And then none of the chum exposed to the tire leachate died either, right? So this definitely looks like stormwater. And then finally, I'm not going to spend time taking apart this figure here, but this is comparing the blood responses in coho. Actually, this is just coho, because chum, there was no change in their blood physiology either. But for coho, comparing stormwater to the tire particle leachate, showing that there's no difference between the stormwater and the tire particles, and that the response to either of those was the same suite of blood parameters: a greatly increased blood concentration, the hematocrit, and then decreased salts in the blood and also pH. All right, so to summarize this, this step that took place right before the science study, we definitely found that chemicals leaching just from tire particles, ambient temperatures, no solvents or anything special like that to grab more of the chemicals, that this water can be acutely lethal to coho salmon, different life history stages. And that at those concentrations, chums, some people say that, chum were not affected. And also showed no change in that in their behaviors or their blood physiology. So these are responses very similar to roadway runoff. We also see through Ed's work that the chemicals in the roadway runoff from tires are in that acutely lethal range for coho compared to the tire leachate. And so we were able to conclude that tire particles did appear to be an important source of the toxicity of whole stormwater. So from there, I'm going to hand it over to Ed, which means stopping share. Okay.
All right. Hello, everybody, and thanks for having us by to talk about our data today. I really appreciate it. I'm going to start to share my screen, and then I'll kind of give a little bit of an intro, I guess. So cool, that should be coming through right there if all's gone well. So I showed up here at UW, Tacoma, and Seattle in the Center for Urban Waters in 2014. And we basically had a really awesome water quality lab. And in fact, that came from some state investments in the Center for Urban Waters, which built some really cool capabilities for the Center for Urban Waters. And I'd always been following chemicals around the environment, especially thinking about chemicals that impact fish. Like that's kind of what I've done my entire career. And everybody was just talking about, you know, the stormwater issue and the fact that there was a water quality linked but unexplained issue going on with Coho salmon. And so I was really lucky and happy that I could essentially join Jen and Nat and Jay Davis and all the other people here who've been really collecting data on this problem for such a long time, and really start to provide some water quality analyses using some instrumentation that hadn't been applied to this problem before. And we started to learn things. And, you know, for several years there, you know, Jen and I were kind of like figuring out new things. And, you know, I was learning what sort of chemicals were out in the field that were present during these types of events. And really the data that Jen showed you was so critical in getting us to a point where we knew we could get a reproducible response in the Coho salmon if we made an environmentally realistic tire leachate mixture. And so all of Jen's work and toxicology efforts that she just showed you got us to the point where we knew if we put, excuse me for a sec. We knew if we put a juvenile coho in contact with a tire tread rubber leachate, those juvenile coho were gonna die with all these distinctive symptomologies. And so really our task at that point, you know, was to understand what exactly was causing that in there. So if you want to read or see, you know, we have a number of publications on some of that earlier work to kind of maybe get us closer to the point at which we could disassemble tire leachate. You know, we have a number of publications on that space, most of which were supported here by, you know, the National Estuary Program and some of the Washington Department of Ecology type efforts. And the technique which we really started to push against this problem was high resolution mass spectrometry. And I'm not going to tell you a ton about chemistry and, you know, analytical type efforts today. I think the big picture theme to take home from this is that the high resolution mass spectrometry techniques are less biased. They're more of an unbiased technique that just tries to detect it all. And then you try and put work to figure out what the chemicals it's detecting actually are, right? It doesn't give you a list of chemical names. It gives you a molecular formula. And from that molecular formula and a bunch of hard work, you might be able to get to identity, right? And I guess what's different about this is it's, you know, you're not making any assumptions about what chemicals might be important. The instrument's just trying to tell you what's present. And once Jen had the toxicology response narrowed down to tire rubber leachate with the distinctive impacts on coho and not chum, we essentially use this in an effects directed analysis or a toxicity identification and evaluation type format, which is designed to disassemble water. Or, you know, chemically, it's designed to chemically fractionate and split all the chemicals present in a complex mixture into components that can then be tested against the coho salmon. So that's really what we've been doing for the last 2 or 3 years here at the Center for Urban Waters. You can see a few of the photos up here. You know, we're packing the tire rubbers into these columns full of glass beads, leaching water through them at room temperature for a day. Again, we're not changing the temperature. We're not putting any solvents in there, nothing like that. It's just like what comes out of those particles. And then they're getting exposed to juvenile coho. the little tanks of 5 Coho, because right now that's the only endpoint we have. We don't have anything like a cellular assay to kind of help us in this process at the current time. So if you think about purple Kool-Aid and you wonder what TIE is, think of it as like TIE just takes purple Kool-Aid that might be toxic, let's say. It pulls out the reds, the blues keep going through, and then you can evaluate, is what came through your column still toxic or not? If it's still toxic, the toxic chemical is part of these blues. If it got stuck to the column, it was a red. And so what we do is we do a similar process just like that with this big complex tire rubber leachate mixture to figure out, as we manipulate the chemicals, where does the toxicity go? And that starts to tell you the characteristics of the toxicant or toxicants. So we did this over the last couple of years. And the key piece here, which again, I'm not going to go into details at any kind of high level right now. If we make that tire wear particle leachate, our instrument typically detects over 2,000 chemicals present in that, right? So here we have 2,200 detections. And then we start just putting it through these different columns and different phases and contacting it with different stationary phases. And each time we do that, the number of chemicals goes down because we're essentially leaving some chemicals on the columns. So you can set up a scheme where you can split, for example, this first fraction into 3, and the middle one, the ethanol elution, is the one that's toxic to the fish. So you keep working with that one. And then this next step of these 4 fractions, it's this one right here, this red one, that's toxic. So you keep working with that. And so once we kept doing that again and again and again, using different types of chemistry over a process that took more than 2 years to optimize. We got to a point where we took a toxic fraction that had 2,000 chemical detections in it down to 4. And at the point you're down to 4, you can start worrying about what their identity is. And by the way, we worried about identity all through this process. We tried again and again and again with chemicals we identified to, we would buy them, we would expose them to the coho. None of those chemicals that we identified along the way killed the coho salmon. So we got a toxic fraction that still had just 4 things in it. And then you see here this little red precipitate. That's what happens if you just take the solvent away. You get this chemical precipitate. And the key aspect of that is it had this molecular formula. The dominant peak in that chemical precipitate was a C18H22N2O2. And it wasn't showing up in any environmental literature or databases we looked at. So it seemed to be a true unknown. And for the last year or two, we've been suspecting we were dealing with a transformation product, which is like you start with some sort of parent chemical and then it reacts with something out in the environment and then it's changed into a different chemical, right? That's called the transformation product. Transformation products are often difficult to work with because you can't buy a standard for this often, or they might not be listed or its structure might be unknown. So they're just a harder place to work. And then last December, Zhenyu Tian really made a key connection. He figured out that this C18H22N2O2 compound that we've been looking at for months and trying to figure out what it was, he figured out that there might be a relationship to C18H24N2, which was the 6-ppd compound, which is used as an anti-ozonant in tire rubbers. All tires break down if they're exposed to ozone. And there's always a little bit of ozone down near the ground, right? There's a little tiny bit of ozone that's very reactive to those rubber polymers. So to make a durable tire, to make a tire that lasts a long time, you need some of these preservative chemicals present. And at least, again, we're not really sure when 6-ppd became highly abundant in the manufacture of tires. We know it was discovered in the early 1950s, and at least by the 1970s, 1980s, it seemed to be probably the dominant anti-ozonant used in tires. So it's been around for a long time. So Zhenyu saw that there might be a link. And again, we all owe Zhenyu Tian a great debt of gratitude. He really did some amazing work. And we found this paper from 1983, which looks at the ozonation of 6 ppd. Here it's called HPPD, but this is 6 ppd. And we saw in that table 1, the C18 H22 N2O2 compound that we'd been staring at for months. And so this really gave us a link between, you know, tire leachate that's killing the salmon, and now a chemical that's used in tire leachate that, upon reaction with ozone, which is what it's intended to do, it forms a product with the same molecular formula as the one we were seeing in the toxic fraction. So we bought some 6 ppd, some industrial grade 6 ppd, we ground it up and we ozonated it. And this is something that Mike Dodd at UW CEE helped us a lot with. He's an expert in ozonation and oxidation chemistry. So we ozonated 6 ppd in the gas phase. The 6 ppd crystals that you see here are this dark purplish brown, right? And you can see this black layer here, right? As you push the ozone through the little pores between this powder, it starts to turn the powder black. And that's the 6 ppd reacting with ozone. Again, the same type of reaction that's happening in the tire rubber, or more likely right on the surface of the tire rubber as the 6 ppd is exposed to the atmosphere. So we synthesized this mixture of ozonation products of 6 ppd. We purified it. Andre Simpson did some NMR analysis that can tell you the structure. We saw that the 6 ppd product that we formed with ozone was exactly the same chemical structure as what was present in the tire leachate fraction that killed the Coho. It came off our analytical column at exactly the same time, and it had the same mass fragments. So again, just like multiple lines of evidence, you know, these are the same exact compounds. The one we were seeing in the tire leachate that was toxic to the Coho, and now one that we generated from a pure standard. And additionally, that NMR analysis gave us the structure. So that's how we know it's 6 ppd quinone. Quinone is, quinone just means like how the oxygens are arranged on this aromatic ring here, right? So this is a paraquinone, the double bond O in the middle of the molecule. And so now we knew the chemical formula, we knew where it came from, and we knew what its structure was. So these were the pieces of information that we had to work with. We got to an identity.
Did you want to say anything about the dinitrone you had on the other slide?
Oh, yeah. Originally in 1983, they presented a dinitrone structure for this C18H22N2O2. They just had less advanced instrumentation, and essentially they saw the same compound, but they assigned the oxygens into different locations in the molecule, right? So they gave it a different name because they thought the oxygens were in a different place in the molecule. Andre Simpson has some of the best NMR instruments in the world. We just have more capabilities now. And so that's how we got to the quinone structure and name instead of the originally proposed dinitrone structure and name. Yeah, so thanks for the clarity, Jen. So yeah, so this was what was there, right? That chemical analysis said this was what was in our sample. So, you know, we tested it on juvenile Coho. And here you see nominal concentrations. That means the concentration you think you put in there. That's not what you measured in there. It's what you expect is in there. If we put in 30 micrograms per liter of the 6 ppd parent, all the juvenile Coho survive at 24 hours. If we increase the 6 ppd concentration, again, this is the parent compound, up to 450 micrograms per liter, which is roughly kind of what is soluble in water. 6 ppd is not very soluble. We saw 1 dead Coho and 1 symptomatic Coho after a 24-hour exposure, right? Now, if we did an exposure with just 20 micrograms per liter of the unpurified mixture, the unpurified ozonation mixture, which is like, we ozonated that 6 ppd powder, that black I showed you, we just took 20 micrograms of that, put it into a liter, you know, essentially. We just used enough mass of that unpurified mixture which contains 6 ppd and whatever was made upon ozonation, which included the 6 ppd quinone and other things, all the Coho died in 24 hours. And then the purified 6 ppd quinone at 20 micrograms per liter, Coho were symptomatic there within 90 minutes. They were absolutely the first ones to become symptomatic, especially relative to the unpurified mixture. Everything just happened a little slower in the unpurified mixture, right? Same behaviors, same look. It was just happening a little bit slower. But these were symptomatic in 90 minutes, and they were, I feel like 3 of them or so were dead at 3 hours, and maybe the 4th and 5th ones were dead right around 4 to 4.5 hours. So that killed all the Coho in less than 5 hours. So that was a really powerful bit of evidence that that 6 ppd quinone structure was highly toxic to the Coho salmon and could kill them quickly. Jen had been doing a bunch of dilutions of either roadway runoff, right? The same runoff she's been working with for a long time, or tire wear particle leachate. This is the dose response curve she got for those. When we measured the 6 ppd quinone concentrations in there, we get an LC50 value of 0.82 micrograms per liter. Again, that's the concentration we expect to kill half of the exposed population at 24 hours, right? By the way, I should point out that 365 Coho salmon were used to make this figure. So this is not, we're not talking about one tank of 5 fish. There's a bunch of replicates in here, and a lot of fish went into making these figures. When you compare that to the controlled exposure, again, we're just adding different concentrations of 6 ppd quinone to the tanks of the fish, and we see how many die and how many don't die, right? The dose response curve. We get an LC50 value of 0.79 micrograms per liter, which is almost the exact same as what Jen saw for the highway runoff and the tire wear particle leachate. So this is essentially the similarity of these values tells us that 6 ppd quinone is the primary causal toxicant in these complex mixtures. It essentially says you can explain all the toxicity you see if you understand 6 ppd quinone. And I'd point out too that the controlled dose exposure on the bottom, each of these, we were doing 10 different concentrations with 8 fish per tank. So each of these runs took 80 Coho. It was replicated twice, right? So that's 160 fish total are used to make this dose response curve here and get the LC50 value. Yeah. And now I just want to say a couple minutes about 6 ppd, right? Like, as a scientist and researcher, you always ask yourself, does my data make sense? I saw these things, can I step back and kind of just think about it and say, like, does that kind of fit maybe with what we expect to happen or what other people have seen? Again, when you're working in a new place, right, it's not necessarily like you have literature support for every observation you make. And so you try to like check it or test it. So what I'm going to use in the next series of slides is, this is all statements basically from EU technical reports by governmental agencies or working groups about 6 ppd. Because again, 6 ppd has been around for a long time. It's been characterized, by governmental agencies who give summaries on, is it a persistent bioaccumulative toxic compound, things like that. So first, 6 ppd is abundant. Passenger vehicles might contain up to about 1% or so of IPPD and 6 ppd. Commercial tires may be up to 2%. So based on the information we find, we put a range of like 0.4% to 2% of the mass of tires seems to be 6 ppd based on what's publicly available, right? Again, when you buy a tire, it doesn't come with an ingredients list, right? So we have no way of like figuring out exactly what's in each of these tires. We have to just base it on some of these broader, publicly accessible insights into tire formulations. 6 ppd is designated a toxic chemical. Again, you see the PBT working group, that's persistent bioaccumulative toxic. says the substance is not considered to be a PBT substance. It does not meet the persistence criterion. It does meet the screening criteria for bioaccumulation and toxicity. And in particular, some of those toxic concerns come from the fact that 6-PPD has acute toxicity in fish at levels something like 8 to 28 micrograms per liter. And I would just point out, remember that the 6-PPD quinone around 1 microgram per liter, right? For roughly 1 microgram per liter. So this is very close actually to the values, within an order of magnitude of the values that are reported for the 6-PPD parent compound. And those are based on, some of the indicator organisms. I think this is Medaka right here, right? This data here is based on exposures to Japanese Medaka, which is a commonly used indicator organism. So again, 6-PPD known to be acutely toxic to fish at pretty low concentrations. So that kind of toxicity characteristic is logical. It's already there in the parent compound. It's also important to note that 6-PPD is unstable. And I kind of think for a long time, this is why people thought 6-PPD was relatively benign out in the environment. It reacts pretty quickly. It hydrolyzes pretty quickly with half-lives of around 3 hours in water. So it breaks down quickly by this hydrolysis pathway. And it's very dependent on like pH and oxygen and temperature and things like that. So 6-PPD is pretty unstable. And so that's why when we did a comparison of 6-PPD, which is in the green over here, it's LC50 with the 6-PPD quinone. By nominal concentrations, again, not by measured ones, it looks like there's an LC50 value maybe of like 250 micrograms per liter for the Coho salmon for the parent 6-PPD, but maybe around 1.5 micrograms per liter for the quinone product. In reality, this is reacting away during the test. So really the curve should be somewhere here in the middle. It's just very unstable with a lot of product formation due to its reaction with water. I note that that's not a reaction that seems to happen for 6-PPD quinone. 6-PPD quinone, based on our observations, is more stable and it hangs around in water for days to weeks without degrading. We're still working on understanding exactly what its lifetime and what its fate processes are. So that's still in progress. And then finally, you'll note from, again, these same reports, 6-PPD, which reaches the surface of rubber products, will be rapidly degraded by ozone or other phytooxidants. The predominant sink of 6-PPD is the reaction with ozone, which is the chemical base of the anti-cracking effect of 6-PPD. So 6-PPD is in tires because it's designed to react with ozone and maybe some other oxidative compounds, but ozone's the important one. So that says if we put 6-PPD together with ozone, we should expect 6-PPD quinone to form. And our data is very consistent with that. When we look in the roadway runoff, we've detected 6-PPD quinone in all the roadway runoff samples from busy highways that we looked at. And notably, when we compare to the LC50 values, all the concentrations that we see there are above the LC50 values, which is very consistent with what Jen sees when she does those dilutions. And she's diluting that roadway runoff 10-fold and 20-fold and 25-fold before the mortality really seems to start to go away, right? And so these types of detections just make a lot of sense from that perspective as well, just looking at this chemical. And then in terms of receiving waters, here you have Seattle urban creeks. The top 3 points here are storms where adult coho were observed to die in the field. That point and that point and that point. So when we're detecting 6-PPD quinone in the rising limb or near the storm peak at concentrations near or above the LC50, that's concurrent with independent observations that adult coho salmon died in that exact storm in the water we were sampling. So it's environmentally relevant, with respect to our detection of it in Seattle urban creeks that have issues with coho mortality. And finally, Jen showed some videos and things like that. This is, I think, my last real slide. Here we have a lower Duwamish mortality event. Look at those behaviors in the adult coho. This, again, there's lots of videos online showing this type of circling, gaping type behavior. And here's our lab exposure where we exposed the juvenile coho salmon to 6 ppd quinone. Oops, hold on, I hit the wrong one. And we just have that exact same observations as what's seen out in the field. And so, there's just a number of lines of evidence which say something important happens when 6 ppd reacts with ozone. The coho salmon, seem highly sensitive to what's going on there. So, I think these observations are clear. I almost feel like I don't even have to summarize them. Coho salmon are highly sensitive to something in those tire rubber leachates. And in particular, that isolated 6 ppd quinone compound seems to explain what we see out in the field. And then especially given the characteristics of the 6 ppd parent, we shouldn't be surprised when you put a toxic compound somewhere and it reacts and it forms a compound which itself has toxicity. The pesticide industry in particular, for like 60 years now, 70 years, has been very aware and studies very carefully retained toxic properties among transformation products, which is very consistent with the observations we see here with the 6 ppd quinone system. There's a famous saying in drug discovery that the fastest way to make a drug is to start with a drug. So a lot of drug discovery just tries to start with a drug and tweak its chemical structure a little bit. And generally that often results in another drug. And that same mindset holds when you think about toxicants. Probably the fastest way to make a toxicant is to start with a toxicant, which seems, again, consistent with what's here. So we need to do some work to understand what's up with these reaction products of ozone and 6 ppd out in the environment. I'd really like to thank my team and all my sponsors. Jen Utian in here put such hard work into figuring this out. We all owe him a debt of gratitude. He was awesome. And then Kathy Peter, whose name you've heard a few times, she built some really cool data foundations for a lot of this work and helped us a ton in just the difficulty of these studies. So I'd like to thank them. And again, many of the same people who are supporting Jen supported us. We thank everyone, especially the Suquamish and Puyallup tribes. We used a lot of their coho for these studies and they were just a huge help. Details are out there and you can talk to Jen or I about any of this if you have questions.
That's just awesome. Nice job, great talk. That's just great. I know there's a couple questions out there. Paul, did you had some questions, and then maybe Ken Warheit?
Right. Can you hear me?
Yeah.
Yeah. So